Hi, folks. It's Brian here from Atomic Ecology. It's 2026, brand new year for Atomic Ecology, and brand new year for all of us, I guess. And I've got a few new offerings coming out from Atomic Ecology this year, and one that I'm really excited about is what we're doing right now is Atomic Ecology Meets. And this isn't a webinar, it isn't a seminar series, it isn't a podcast, but hopefully it's something that combines all of these, the best parts of all of these. Over the course of this year, I'm going to be meeting with a host of different leading lights in our field, various different isotope scientists, isotope ecologists, and we'll be discussing some of their new research, some research that they're really excited about, but these are going to be shortish presentations, 10, 15 minutes, 20 minutes maybe. And then we'll have a bit of a chat about the story behind the research and the story behind the researcher as well. And my hope is that it's going to give people a better understanding of what we're doing, what can be done with these wonderful little ratios, and the people who are doing it. I had a great opportunity just before Christmas to sit down for an hour or so with Dr. Sora Kim, and we discussed some of her work on sharks, shark behavior, shark isotopes, and Sora's journey towards trying to determine trophic fractionation values and isotopic incorporation rates for species that are very difficult to figure these things out for. So, without further ado, I'm going to stop now, and I really hope you enjoy this first episode of Atomic Ecology Meets. Welcome to the first of our Atomic Ecology seminar series, and I'm super excited to be joined today all the way from Germany by Sora Kim. Sora, I've known for a couple of years. We worked together on an isobank project in the US, and then she very kindly accepted an invitation to come and talk to us all at IsoEcol in New Brunswick a couple of years ago. So I'm super excited to have Sora here to talk to us about some of her work today. For those who don't know her, Sora is an associate prof of ecogeochemistry at the University of California, Merced, a place with, I guess, a short history, but a long history of stable isotope research, yeah? Where Sora's also serves as faculty director for their isotope lab there, the Stable Isotope Ecosystems Lab. Sora's background, she started with a BA from Dartmouth College, focusing on earth sciences and environmental studies, and subsequently moved to a more, well, scientific qualification, at least, in getting a PhD from UC Santa Cruz in Earth and Planetary Sciences. And Sora's joins us today from Germany, where she's taking a sabbatical from what I think is one of the most exciting and interesting isotope research projects or careers out there. Whereas Sora's career really sits, as I see it, almost in a nexus between ecology and geochemistry, looking at isotopic tracers to understand feeding interactions, biology, ecology, but predominantly of really old things. At least that's where I got to know Sora from. But as far as I'm aware, today she's not going to be talking about really old things. So with that, welcome, Sora. I'm going to quiet myself and slip into the background, and I'm going to let Sora take over, share her slides, give us her short presentation, and then we'll get back afterwards. We'll have a little chat about the research and the researcher as well.
So I was trying to figure out what to talk about, and I thought that this was the right stable isotope nerdy crowd that I could get away with this talk. So I titled it Stable Isotope Lessons from Captive Leopard Sharks. I think that the audience is either isotope curious or already isotope experts or soon to be experts. And so in thinking about how do we actually interpret our data, we oftentimes rely on these feeding studies and the biological parameters that come from them. So I am interested in a lot of shark ecology, both in the modern and the paleo. And um, I chose my technique to be stable isotope analysis. As an undergrad, I became interested in stable isotopes. But then in grad school, I started working on sharks. And to date myself, for better or for worse, I graduated in 2010. So that would be about right here. And if you look at the line for stable isotopes, this light blue line, it's very low. And so when I first started, stable isotopes was really, I think there were 5 papers or something like that with sharks and stable isotope analysis. If you did a Web of Science query, because we didn't have Google Scholar then. Um, but now it's actually much more increase, and it's on par with stomach contents as the predominant way to look at diet and habitat preference. And sometimes I meet people who are modern day shark ecologists, and they look at me and they're like, "Oh, you're the reason why I always have to take a biopsy or a blood sample for stable isotopes." I'm like, "Yes." So yeah, so I'm going to talk about this captive feeding study that I geeked out on and still geek out on pretty regularly. So, organic carbon and nitrogen isotopes, they vary based on biological, physical, and chemical processes, which probably most people are aware of. There's these baseline variations, and I call these spatial variations. You can see them here with this chlorophyll concentration map in the Northeast Pacific. With chlorophyll concentration, red at the northern latitudes and near shore has the highest chlorophyll, and then sort of in more equatorial waters, we have the lowest chlorophyll concentration. And that actually scales with also gradients in isotopes, both of carbon and nitrogen. So we have a latitudinal gradient as well as nearshore, offshore. And and then sort of what we're more used to thinking about as ecologists is this aspect of like trophic level and food web dynamics. And so we think about spatial variations usually in the realm of carbon. But then for trophic ecology, we think about nitrogen isotopes. And here we have trophic level and it increases with delta N15 values. Let's see. And to interpret stable isotope results, you need 2 biological parameters, trophic discrimination factors and incorporation rates. And so you can stick almost anything into an EA that has organic material, and you're going to get a delta C13 value and a delta N15 value back. And hopefully they're good values. But what you can actually say with those numbers is dependent, if it's coming from an animal, it's really dependent on these 2 biological parameters. So when I started my PhD, the Monterey Bay Aquarium was actually keeping white sharks in captivity, or in the aquarium. And I knew that that was an impossible task because the Monterey Bay Aquarium couldn't even keep white sharks around. But I thought that maybe I might be able to hang with keeping some leopard sharks around. And so usually, shark ecologists and Shark Week, they're all interested in the charismatic megafauna like white sharks. But sort of for study organisms, sometimes we have to scale down. So we'll be talking about leopard sharks today. And if you look at trophic discrimination factors, this is really a cumulative fractionation effect of metabolism for an organism. This is a compilation by Post in 2002 for invertebrates and vertebrates. The top panel is for the trophic discrimination factor of nitrogen. The lower panel is trophic discrimination factor of carbon. And these are for all kinds of tissues. But basically how we come up with these numbers is that we measure consumers' tissue while it's in steady state with a controlled diet. And we express this oftentimes in the big delta notation, but oftentimes folks these days are just calling it a trophic discrimination factor or TDF. And incorporation rates also vary by species and tissue based on the diet and metabolic physiology. So here I'm showing some data from Hobson 1992 with quail. And what I have plotted is for muscle tissue, but they've measured a bunch of different tissues like blood, muscle, and bone collagen. And you can see sort of the metabolic activity of these different tissues determines what the incorporation rate is. And so this is the rate that dietary carbon and nitrogen are assimilated into tissues. And to solve this, you switch between two isotopically distinct diets and monitor the isotope values of the tissues throughout the switching period. But there's some special considerations for sharks. And I oftentimes speak about sharks as deeply cool, but very weird. So what people know about sharks are that they're carnivores. Most of them are ectotherms, although we do have the lamnids, which are partially endothermic. Sharks also retain their urea, which does funny things in terms of nitrogen. And then they also have intestinal valves that maximize nutritional absorption. So there's almost 500 species of sharks, skates, and rays out there, and they occupy, everything from the benthic to the pelagic, nearshore, offshore, the abyssal plain. They are really prolific within our oceans today and have been around for 450 million years. So getting a sense of what they're doing would be cool, but we have these physiological parameters to take into consideration. So I designed a feeding study. There were only 9 individuals, but they are very well studied leopard sharks, Triachus semifasciata. The first part of the experiment was to equilibrate them to a squid diet. So that was the first 565 days. And then for the remainder of the time, and the experiment went for 1,200 days, 3 individuals, they continued on the squid diet as the control, and then 6 individuals were switched to tilapia, which I sort of deemed my experimental group. And the tissues that I collected were plasma, red blood cells, muscle, vertebrae, and teeth. And this is an image of a leopard shark from Monterey Bay Aquarium, and then various images of us sampling leopard sharks. So I'm going to review some of the broad brushstrokes of the data, but I'm going to kind of dig into it in sort of dimensions that most people, I think, oftentimes skip over for this data. So one of the things I did was I calculated these trophic discrimination factors. And oftentimes, people just look at this number of the big delta N15 or the big delta C13. But I would like to draw your attention to that there's actually a lot of variation among the individuals in terms of isotope composition. These are only the control individuals, but I want you to look at this X bar at the X axis, and we go all the way out to day 1000. And so I'm not even counting, the first, say, 600 days of the experiment where they're coming into steady state with this squid diet. If you just look at, the last 400 days or so, you can see that variation decreases among the individuals, but there's still some oscillation that's happening both in terms of the plasma and the muscle tissue. And then, like I said, for estimating incorporation rate, it requires this isotopically distinct diet. So the squid we got with this steady state with the squid diet. But then if we look at incorporation rate, these are the 6 individuals that were switched to tilapia. And again, you can see that there's a lot of individual variation. And so you can, oftentimes just express the mean of all the individuals, the mean and standard deviation, but I'm kind of purposely showing all the variability that's within the system here. And then we also looked at teeth. I'm interested in the fossil record, and so teeth is really what I was going after in designing this experiment. And what we found, you can see this in this top panel here, is that the incorporation rate of the teeth, that's the very last bars, they are actually similar to the plasma. So that makes sense that the nutrients that are going into the teeth to build the collagen in the dentin are coming from plasma. The other thing that we were able to see is that they are actually chronological recorders. And so here we have that same calculation of tissue incorporation rate, but you can see this really long delay here. And that's a 250-day delay in the signal from tooth formation. So here I have a picture of a shark jaw. This is not a leopard shark because leopard sharks have really tiny teeth and it's hard to see this. But the teeth, they form in the back and then they slowly move up to the front in sort of a conveyor belt fashion. And so we're just clipping teeth from the front. And so it takes, 250 days for that tooth to go from formation to the front of the jaw. Uh, and so that's all good. And this is another tidbit that I think that people often overlook about this experiment is that the trophic discrimination factor actually varies with diet. And so here again, this is the muscle tissue that I'm showing with days after the diet switch with delta C13, all that variability that's going on among the individuals. And so with this switching experiment, I was able to also estimate what the trophic discrimination factor would be for carbon and nitrogen on the tilapia diet. And so here I'm showing you a table of plasma, red blood cells, and muscle tissue, and then a side-by-side comparison of the trophic discrimination factor between these two diets. And hopefully you can see that they're different. I don't have statistical tests on these, but for some of these tissues, they're very clearly significantly different. So what's going on? Another study that we did, and this was led by John Whiteman, was a look at compound-specific isotope analysis for clues about these trophic discrimination factor differences. And so we have carbon on the left, nitrogen on the right. Both of the top panels are the delta C13 or the delta N15 values of the individual amino acids. And then the bottom row is the offset between the shark and its diet. And so we again show the control sharks and the experimental sharks. And in this pink box, I've highlighted the essential amino acids. So for carbon, we think that essential amino acids, are coming directly from diet and that they see less transformation. And for the most part, you can see that there's overlap between the diet, which is the open box, and then the shark in this top panel. But for the experimental sharks, there is more of an offset. So what's going on there? Tilapia is not a natural diet, so that's one possible explanation. But then you can see here for this offset between shark and diet that for the control sharks that they don't quite sit on this line of zero, but there are some amino acids that do. And then the experimental sharks are definitely off of that zero line. And then for nitrogen isotopes that we have these source amino acids as well as trophic and source amino acids. And you can see that they line up kind of for some of these amino acids, but actually for lysine and tyrosine, that there's actually quite a bit of difference between the control and the experimental individuals. And then again, if you look at the offset between the shark and the diet, you can also see that there's quite a bit of offset there. So what could be going on? One idea that's been given before with captive feeding study is that trophic discrimination factors vary with isotope composition of diet. And that is not what's going on here, actually. And so this was really quite slick. John came up with this. And I think that this is really important to consider. And so if you look at the difference in the delta C13 value or the delta N15 value of the control diet to the experimental diet, and then the offset between these two diet items, you can see that they are linearly related, okay? So this is, what is the offset in the consumer and what is the offsets in the diets? So TDFs, they're not actually varying solely from the isotope composition of the diet. It has something to do with, this offset between the pre and post diets of the diet switch. I do want to give like a small amendment of saying that while TDFs don't vary with diet, with isotope composition of diet, that there can be contributions of gut microbes to the essential and source amino acids. And so there is possibility that those don't exactly line up with zero, and that's why. But TDFs aren't actually varying with what the diet isotope value is. Instead, this is the explanation that we have, is that larger differences between the diets in a diet switching experiment, they are magnified when you have a longer turnover rate. And so in this top panel A, you can see in the dashed lines the difference between the new and old diet. And when you are tracking the consumer, which is this black line, whether you're taking a measurement at 100 days or 50 days, the error is actually not as much. But then if you have a bigger separation between the new diet and the old diet, whether you take your measurement of a TDF at 100 days or 500 days, that magnitude actually shows up much more. And then if you have something like in this case with the tilapia and the squid diet, where they have really drastically different diet delta C13 values, then that's when you really get a much bigger emphasis of that variation. And so this is where it's really important that you can see that like at 100 days, that's this D panel over here. If you are not going out to steady state, if you are taking your measurements of TDFs, they can be really a lot of error associated with it. Whereas if you are taking your TDFs at 500 days when things are absolutely at steady state, it does not matter what the isotope composition of the new and old diets are. So the conclusion of this study with John and I think Seth and Kelton were also on this as well, was that the muscle tissue turnover is actually less than 50% for nitrogen. And this is after consuming tilapia for 685 days, which is a little bit frightening and difficult, and means that probably no one should ever do another captive feeding study with sharks again. But this was, I think, a really good sort of application of compound-specific isotope analysis to give us a sense of what's going on metabolically for these sharks. So one last thing that I'm going to end with here is looking at eye laminae. These are eye lenses, and I am really excited about this work. It is by my recent PhD student, Dr. John Kuntz, and eye lenses are this novel accretionary substrate for stable isotope analysis. So he caught leopard sharks, and I will show you the data from the wild leopard sharks they caught. But from these eyeballs, this is what it looks like here, but you can peel off layers of the laminae, and the diameter, is it scales with the age of the shark. And so he peeled back all of these layers. And then he used the TDFs and incorporation rates from the vertebrae to sort of assume and extrapolate for the eye lenses. So essentially, he's calibrating TDFs and incorporation rates for the eye lenses based on these feeding study results. So in panels A and B, you can see the offset between the eye versus the vertebrae. And so for carbon, it's quite flat. It's a very steady offset. For nitrogen, there is a little bit of differentiation due to the composition, the protein composition of the eye lenses. But then when he compares sort of the what do you call it? The pattern that you get from eye lenses versus vertebrae, you can see that they really do mirror one another. And he's used a couple of different methods to sort of line them up to do the wiggle matching. And then he's also estimated what the island's trophic discrimination factor would be if you make assumptions based on offsets between muscle and eye lenses or vertebrae and eye lenses using nearest neighbor or dimensional time warping, I think is what DTW is, which sounds really cool. I did not do that math. John did it all. And this is for the wild specimens that we caught in sort of the entire records from eye lenses. And so let's see here, the top, we have carbon isotope values, and you can sort of see that with ages that delta C13 values vary. He's able to determine this sort of embryonic time. And the embryonic time lines up both in terms of carbon and nitrogen, and it really suggests a shared nursery. And then you can see after the embryonic time that these different localities that they actually have distinct isotope values. So it almost suggests that, or what we're arguing is that there are subpopulations within the Central California leopard shark population. And so we have these sites of the lower South Bay, which is down here, and then in this bottom right-hand corner, it's the yellowish colored line, and you can see that that is very distinct, but overlaps with all the others. And then we have in the north, we have Drake's Cestero, Tomales Bay, and Bodega Bay. And these areas all have differing amounts of human impact. And so this is something that's in review for a special issue to commemorate Keith Hobson. So just in closing, just wanted to say that leopard sharks, they're sort of this model organism for shark-specific trophic discrimination factors and incorporation rates. So far, the newest tissue to be analyzed is eye lenses, but we've looked at vertebrae, fin cartilage, teeth, muscle, blood plasma, and red blood cells. John has made this cool figure, and it wasn't until he sort of showed me this figure and also kept referring to leopard sharks as this model organism that I was like, oh yeah, you're right. They kind of are. It's kind of cool. He has really done a lot to sort of take what I did for my PhD and then take it into a new realm, which is very exciting. So with that, I would like to thank you for coming to the Cool Shark Show with this awesome piece of art by Ray Troll, and have various acknowledgments of people that helped with the various projects, as well as different funding sources.
Hi, folks. It's Brian here. I hope you enjoyed that presentation from Sora. Before we get into our little chat, I wanted to, no such thing as a free lunch, I wanted to highlight a bit about how what Sora was talking about is covered in some of the courses that are available here through Atomic Ecology. And particularly, Sora's presentation really highlights how key and how important it is to know trophic fractionation values and incorporation rates if we want to be able to infer consumer ecology from isotope values. And this is an aspect that is front and center in my Essentials of Stable Isotope Ecology course. And if you have not already signed up for this course, I strongly recommend that you do, because one of the modules in this course focuses entirely on this concept of isotopic incorporation. I talk a lot about trophic fractionation. We've got a whole video in here on that, how to measure it, how to infer it, and what we can use to estimate it if we don't have known values. And similarly, there's an old topic in here, another lecture in here on tissue turnover rates and how we can incorporate those into our ecological interpretation of stable isotope data. There's a lot more in the course, but I've got other stuff out there recommending that or talking about that. So I'm not going to take up any more of your time. And with that, let's get back to Dr. Sora Kim. Fantastic. Thank you, Sora. It's a shame that we don't have a room full of people to applaud. That's the one part of this that I couldn't quite figure out how we were going to match, you know?
You can do the fake
I can
applause.
do the fake, yeah, I can hit my
I'm
little
sure that
icon down here.
ChatGPT can come up
I'm
with
sure
fake.
something will we'll find it somewhere, you know, but
Yeah,
you can imagine, you can close your eyes and visualize it, you know?
yeah,
Yeah.
yeah.
Fantastic. That was brilliant. And so
Super nerdy,
yeah, yeah, yeah.
like
But
a deep
it's
dive
great that
into
when we have this
isotopes.
what can I talk about that I'm never going to have to try and pitch or sell? I could just get to talk about it, you know?
Yeah,
And it's strange. I find this with myself and with a lot of others that we always end up going back to our PhD projects for these things, you know, that's there's something happens in that time that it's still, I don't know, well, maybe it's to do with the 1100 days
yeah,
of captive rearing in order to get your data. I can only imagine
yeah.
the stresses and the strains associated with that, you know?
Yeah, they required, I think I couldn't go home for Christmas 1 or 2 years because I needed to
You
like
needed
take
to be
care of the
there.
sharks.
Wow.
There was
That's
there was a time that we were worried about like animal welfare
yeah.
protesters coming in
Oh no, my
and
goodness. Yeah.
harming the sharks. So I might have like slept by the shark tanks.
Literally sleep with the fishes, you know? Yeah.
So yeah, I mean, I don't know if you've ever been to Santa Cruz, but Highway 17 is this pretty windy uh,
Okay, yeah.
mountain road. And so when I brought the sharks to UC Santa Cruz, I drove across that mountainous road with a tank strapped onto a flatbed truck and then like an oxygen tank in the passenger seat buckled in with like an air hose bubbling in. And now I think about it, I'm like, I wouldn't, that
Yeah,
seems
yeah,
like
yeah,
such a bad
yeah,
idea.
yeah. No, my brain is going to the that final scene in Finding Dory, isn't it? Where they're they're bouncing around at the back of the truck. Yeah,
Totally. Yep.
yeah, yeah. But it worked, you know, and you got really cool data out of it. Yeah,
Yeah, and I mean, honestly, like it's a N of nine, like I
yeah,
said. But
yeah.
at this point, I think that these sharks have been featured maybe in like 12 papers or something.
Yeah, yeah. Well, and rightly so, you know. So I've got a couple of I do have an actual researchy type questions as
Yeah.
well as just sort of blubbering. But what was I going to say? Yeah, so two things, all right. The first thing is so you're collecting you mentioned a good few times that you're interested in teeth and you showed this really cool really long lag of 250
Hmm.
days and then an immediate shift
Yeah.
from the teeth. I'm starting to do some more work, or I have been doing some more work with archaeologists, and then they talk about teeth, and I kind of get scared because it's inorganic carbon, and how do you
Hmm.
understand it? How do you get data that you can compare with soft tissue, with muscle and keratins and things like that,
Hmm.
that I'm more comfortable with or familiar with? What are you, how are you using these teeth to get at real ecologically significant values, say, or something that's
Yeah.
meaningful?
Yeah. So when I work with teeth, these teeth were actually too small. But with larger teeth, I actually drill out the dental collagen so that
Ah, okay.
a lot less inorganic carbon to
Yeah,
worry about. Like there is still some mineralized, um, carbon from the carbonate that you're
yeah,
talking about, but I
yeah.
usually try to drill teeth so I don't have to worry.
Ah, okay. So you're getting the teeth, but then you're just taking out this collagen component
Hmm.
and doing your analysis on that
Yeah.
then. Yeah,
And so
that
for
makes
these
sense.
teeth, I think that I probably did react them with some dilute HCl
Yeah,
so that
yeah.
I could try to I
Just
like probably
remove
crush
any of
them
that.
and get rid of the
Yeah.
carbonate. And then, the dilute HCl won't actually affect the organic
Yeah.
the bound amino acids that are within a protein structure. That usually requires higher HCl
Mm,
concentration. And so I think that most modern shark ecologists don't look at teeth, but
mm,
I've actually think that they are really cool structures to look at, even in
mm.
the modern, because they give you this sequential
Yeah.
time frame.
Yeah.
And so some of the projects that we've been doing more recently are to actually analyze teeth from the different rows of
Yeah.
the shark, and then to be able to say, like, seasonally, is the shark diet actually varying? And so some sharks that they can move around quite a bit, obviously, and then some are
Mm.
more local and endemic. And so if they are local and endemic, it might be that the baseline could be changing through the
Mm.
seasons, like with upwelling or something like that.
Mm-hmm.
Or it might be that there's actually different kinds of prey that are more abundant,
Available.
and therefore their preferred prey. And then for the sharks that are moving around, it could be that that type of data I think is best if you can couple tagging data,
Yeah.
either satellite or acoustic tagging data, alongside the isotope data. And so I think that this is where you can't blindly just apply stable
Mm-hmm.
isotope analysis, that you need to have other outside information and inferences and techniques to inform how you interpret it. But then you also need to know enough about stable isotopes that you're not like wrongly making assumptions
Mm-hmm.
in how you interpret things as well.
Yeah. No, that's words to live by, you know?
Oh yeah.
Uh, um, so one of the things I'm really keen on is this individual variability. Yeah.
Yes.
And that teeth in rows, I only learned about that recently. Well, I knew that they grew in rows, but I knew that they were constantly growing. But the idea that you could pick up these rows and go back through
Mhm.
time is super cool. But I'm also interested in the individual variability in the other data that you showed
Yep.
that you see, and you highlighted it, and kind of that it's one of the things I don't know whether it's a change as you get older in your career, or that there's a change in science where people seem more interested or more willing to investigate the noise nowadays,
Yes.
you know? What do you think is behind that? Why are these individuals where you've restricted so much, they're not going to different areas, they're all on the same diet, in the same environment for the same time? Why are we seeing these reasonably significant, like certainly
Yeah.
statistically significant, but ecologically relevant levels of individual difference in the isotope values?
Yeah. So I'm not a huge fan of anthropomorphizing animal.
Mm-hmm.
But I do have to think about, in terms of like our metabolic physiology,
Mm,
we're all individuals and really different, and so I expect that to be the same for these sharks. And so I actually, was either I took both the weight and the total length of these
hmm.
sharks, and so the growth rates vary between all the individuals, but that does not explain actually the variation in incorporation
Mm,
rates
hmm.
among the individuals. But we spent a lot of time with these sharks. So they actually had different personalities and how much they moved around, and like whether they would, I mean, leopard sharks are pretty sedentary. They're
Mm.
like the kitty cats of sharks, I would say. And so some of them, they just like laid on the bottom and like, I mean, it wasn't until the food was like directly at the tip of their snout that they might eat it. Whereas other ones, while we like threw the food in, it just like went up and like immediately wanted to eat it.
Yeah.
And
Wow.
so
Okay.
we do have logs of how much every individual ate with every feeding, but that's a lot of data to go
Yeah.
through. So I
Yeah.
never
Yeah.
did.
Ah.
And we just made sure that, that they were eating at least, I think it was like 40 grams per feeding session,
Yeah. Okay. Yeah.
week. And the other thing that we were able to track as well for these sharks, what when we looked at them, we would look at their gill slits and then every I think it was a few months, we actually had to put them into um a freshwater dunk to get parasites off of their gills. And
Ah,
so they were getting parasitized.
parasitized.
And so,
Mm,
there are other like biological factors going in
yeah,
to the health of these animals as well. So, I think that there's an aspect of, yeah, like there's just different physiologies, but then there's also like different activity levels, and then also uh, potential other effects of parasites or
yeah,
sickness,
yeah.
whatever.
As, as some good old friends are want to tell me that there's a lot of different ways to get to your isotope value, you know?
Yes.
Yeah, and it takes a long time to tease them all apart. Yeah.
Yeah, and I
Yeah.
think that this is one of the things like people oftentimes refer to isotope signatures, and I oftentimes
Mm.
think about a signature as like a unique identifier for
Yes.
a person. But because there are these multiple ways of actually getting at the same isotope value, I actually think that
They
like
get.
maybe they're isotope signals, but
Mm.
isotope signatures is is a little bit further than I think that we
Yeah,
can
yeah. We're
really
comfortable
look at.
going just yet,
Yeah.
you know?
Yeah.
Yeah. Cool. So thanks a million. I'm going to, like I've been saying, we sort of Sora and I had a little chat before we started here, and one of my goals with these seminars is to talk about the science, but also to talk a bit about the scientist and get to know a bit about the people doing this work and what motivates them and what are as because one of the things I've loved about isotope science is the sense of community.
Yeah.
And it's always been a real open, engaging community. Something to do with the size of it as well, maybe. Or every we're all reliant on these highly complicated pieces of infrastructure and equipment that don't always work. So we all have to. It has a way of humbling you pretty quickly, I think.
Yes.
And so I want to take couple of minutes just to chat a bit about where you're at, how you got there, and what your hopes and plans are for the next step. And I mentioned that you're in Germany at the moment, not in sunny Merced. So how's that going, and how are you finding it, basically?
I actually think that Germany might be sunnier than Merced right now because
Ooh,
there's this inversion
controversial.
here
Yeah.
in Merced and it's very gray and
Yeah.
I haven't apparently the sun hasn't shown in like weeks. But
Ah.
yeah, I guess to start, I got involved in stable isotopes as an undergrad, which is I think
Mm.
kind of odd and rare. Uh, I worked in the theater with someone named Jake Wallbauer. Um, who took me to a grad student party. And Jake's mom is Noreen Taras.
Oh wow.
So, yeah. And then I went to this party and I met someone named Paige Chamberlain and a bunch of grad students that
Yeah.
also work on isotopes. And so yeah, I kind of like fell into it.
Nice.
And then after college, I actually helped out Jake to help reestablish Paige's lab at Stanford. And so I
Mm.
got to like actually work with the instruments and like get to spend a bunch of time with them. And I found that I really actually liked that. Like I
Mm.
worked in the theater, but I worked building sets and like helping yeah, behind the scenes kind of stuff. So I like
Yeah.
tinkering a lot.
Yeah.
And so that's also very important because like in the stable isotope world, like we don't do service contracts usually, as
Hmm.
like, it's usually the PI or the lab director that's in there,
Yeah.
like doing the micro plumbing of whatever instrument that it is, and troubleshooting electronics. So I think that that appealed to me. And then the other part of it was just how diverse the different applications were.
Mm.
And so even within my group now, I do modern and paleo shark work, but we have stuff going on with modern and historical wolves, but then also
Mm.
going back to like environmental reconstruction for dire wolves, which were around when like saber-toothed cats were around and stuff,
Very
which is
cool.
kind of rad. Um, we've had projects with soils, projects with archaeology. I've done projects with spiders and birds. I mean, like you name it, and I've pretty
Yeah,
much like stuffed it
shoved
into
it
a
into a mass
cat.
spec somehow, you know?
And so that really appeals to me. Oh yeah, I've
Hmm.
done spats were cool.
But ooh, that's yeah, no.
Yeah.
Well, one of the things when I was in New Brunswick at SynLab, that was the same. That there was just everything you could imagine. We had dripping seal jaws were sent in at one point. I did not. I'm not one of the tinkerers, so
Mhm.
I was very fortunate to be able to just sit back and not be allowed to touch any of the fancy machines, which is probably somebody saw me coming. Let's just put it that way.
Well, I think that once you get to the point of being faculty, like the
Yeah.
reality is like the time for tinkering is much,
Yeah,
well,
yeah,
it depends. There are some faculty that actually are still tinkers. They are really specialized method development
yeah,
type of folks,
yeah.
in terms of the analytical side. And then there are folks that maybe like for me, I would say I'm much more of like a methodological or approach like tinker at this point now of
Yeah,
like what are the
yeah.
new tissues we can do? What are the new ways that we
Mm,
can like apply questions to look at ecological concepts or theories in shallow and deep time?
mm.
Um, so yeah, I think that some people do still have time to be in the lab tinkering,
Yeah,
but it's not me either
yeah,
anymore.
yeah. I hear you. I hear you. And
And then your last question as to what I'm doing in Germany,
yeah.
I am actually working to develop a trophic level indicator that we can use in paleontological specimens. And so specifically, this is mostly for zinc isotopes.
Mm,
And so zinc is something that is preserved in the enameloid of shark teeth, but then also an enamel for mammals as well. But I'm interested in the shark teeth. So this is the part of the project I was doing this time was really getting a lot of data for modern sharks, both
hmm.
from my captive feeding study again. They never yet again, and then also from some aquarium reared sharks and then some wild specimens as well, and then also looking at some fossil localities and getting pilot data. So it's been nice to get back in the lab, and I've done
Yeah, yeah.
a lot of pipetting, which is kind of fun.
Well, I'll take your word on that one. Yeah.
I enjoy lab work.
Yeah. Right on. Um. Oh, there was something I was about to say, and then it slipped my head. So for the uninitiated, what's different about zinc isotopes compared to like a lot of the people coming here will have no, oh yeah, we've done carbon and nitrogen and maybe hydrogen and oxygen and sort of
Mhm.
put it all together. Zinc, I've seen a couple of papers again from that sort of paleo type of realm that have shown it. What's involved in getting those measurements? It's not the same process,
Yeah.
I'm assuming, yeah.
Yeah, so for zinc isotopes, the zinc is actually it's a mineral bound. And so it for delta N15 values in organic tissue, it just doesn't preserve in the fossil record. And so
Mm,
for zinc isotopes, it's actually within the hydroxyapatite structure. It's zinc is an essential nutrient and it's used in an enzyme for
mm,
some cellular function. And so that's how it's incorporated. Um, zinc is, I think it's delta 66 zinc, it's 66 to 68. And it actually decreases in value with trophic
huh,
level from what we can tell so far. So it's just kind of like kind of
yeah,
flip on its head in terms of like how the fractionation
Jim
processes
works.
are working. um, we don't totally know the systematics of like how it's worked out, which is why I'm doing all these
You're here,
model samples. And let's see here. Yeah, so we're also comparing zinc to mineral bound nitrogen. And so that's also a new paleo trophic level indicator where when things mineralize, they actually put out this protein scaffolding to that is where the biomineralization initiates, and then the protein scaffolding is reabsorbed into sort of the matrix. But there are tiny amounts of that organic matrix that get trapped in the
Hmm.
biomineralization. And so I think that yeah, so these are two different trophic level indicators. I haven't done the prep for the mineral bound nitrogen, but it does require it relies on the denitrifier
Right.
method.
Mm.
And then your other question, oh yeah, was the zinc isotopes the prep? You actually have to dissolve the teeth and then put them through a resin column, and then the
Oh,
resin
wow.
captures the mineral portion, and then you sort of like elute out everything
Yeah.
else, and then release the zinc from the resin with
Resin.
nitric acid.
Wow.
So it's a resin
It's
column
a
process.
process. Yeah,
Yeah,
yeah. Yeah.
it's more time consuming
Yeah.
than sort of the bulk isotopes. And I don't think that modern ecologists would really ever have a need for
Need to it.
using zinc isotopes. Maybe if you're interested in some like special metabolic
Yeah.
process or something like that, but it's really for the paleo record for us
Yeah.
to look at like food web studies and things like that.
That's that's super cool. All right, cool. Taking a bigger view here before we're gonna wrap out in a moment or two. But for you've told us a bit about your really cool serendipitous almost route into isotope ecology. For folks who are listening in on this or reading a bit about this, about isotope stuff, or are interested in getting into or applying some isotope work to their research or building a career in it, give them any words of wisdom, thoughts, or advice for a young
Mm.
budding, don't even have to be young, I guess, but a
Yeah,
budding
anybody.
new isotope person. Yeah.
Yeah, I would say send the email,
Yeah.
just cold email folks and ask questions. There have been a lot of people who have emailed me over the years for various shark-oriented stuff, usually. And I always try to do my best to help them out, or and for a long time, people kept saying that they wanted to do captive experiments with sharks. I was like, don't do
You
it.
don't.
And
You think you
it's
do,
a
but you
very
actually
small
don't.
reason. Yeah, don't do it. Um, but then, in terms of like methods and prep and how to
Mm,
assess like your data to see like, did you get all the urea out? Did you get all the lipids
mm,
out? Like, what can you do to resurrect your data? Like, who should you talk to like get that next level of analysis in? Like you said, the isotope community is friendly.
yeah.
And I think what's really different about isotope ecology versus other sort of subdisciplines of ecology or biology is, while we go in with a hypothesis and we have an idea of like, the general question, oftentimes our experiments are much more exploratory,
Mm,
and I think that that's like a cultural difference of
mm,
isotope ecology, and almost all PIs, like for me, I get emails, and maybe I should not say this, but I'm oftentimes like, if it's a cool question, if it's something that
mm.
I think hasn't been done, or that I'm just like a side project that I'm curious about, I'm like, yeah, send some samples, like
Let's
let's
just
run
do
it.
it. Yeah.
Yeah.
Yeah,
It's
exactly.
it's it's
Yeah.
not that
Do it first,
expensive.
ask questions later,
Yeah, and I think that that's kind of like this exploratory spirit, and that comes, from the the folks who kind of initiated
Mm,
stable isotope ecology. I mentioned Keith Hobson, but Marilyn Fogel, who I worked with when I was a PhD
mm,
student, but was at UC Merced for a long time before I went there. Noreen Taras, um, Kate Freeman. I mean, there's a lot of folks in that category,
mhm.
but you know, the I I feel like that the folks who started isotope ecology were really like they were okay with it, just exploring and being curious
Hmm.
and seeing like where the data might take them. And I think that that's really different. Like you don't have to have like an entire experiment and your N of like 200
Hmm.
specimens all like planned out for somebody to like have a conversation with you. I don't know. That's at least been
That's
my
no,
experience.
that's perfect. I can't think of a better sentiment to close out on, to be honest.
Yeah.
So with that, yeah, I'm going to thank Sora again for her time, and you heard it yourself. Send her emails. She wants more emails. So if you're going to regret
Oh man, I might
anything from
regret
today, what did I just say?
Yeah. Well,
Okay.
yeah, thanks, Brian. And
Yeah. No.
I'm super
Fantastic.
excited for atomic energy or ecology.
Or whatever it's called. Yeah.
Yeah, because I know that your videos, your YouTube video series were so helpful for so many folks,
Yeah.
you know,
Yeah. Yeah.
from
No,
COVID until recently.
until
So
right now.
excited
Yeah. Yeah. Yeah.
to
Well,
see what happens
there'll be
next.
a bit more on that soon. Yeah. Cool.
Cool.
Hi, folks. I hope you enjoyed that at least nearly as much as I did. You'd be doing well if you enjoyed it as much as I did, let's be honest. As Sora mentioned at the end there, Atomic Ecology is up and running now. So if you're watching this, hopefully you've already been on our website and you can see some of the various different consulting services that are available, project development, applying isotopes, data analysis. Things aren't working, I can help. And also a lot of training. I'm offering sort of one-on-one group sessions, things like that. Get in touch if you want to do it. Or some online courses that I kind of mentioned earlier. And these fall into two categories: the essentials of stable isotope ecology, which really covers a lot of the theory around things you need to know if you want to apply stable isotopes and not realize at the end of your project that you've made some horrible mistake. And also a data analysis course, which has been very, very successful in the past. Some of you who are coming to the site have probably taken this where it was previously available for free online. Unfortunately, that's no longer the case, but it has been massively updated and improved. And you can come in and sign up here, take any of these courses or get in touch. And yeah, enjoy 2026. All the very best. Bye bye.